It's hard. I mean, it's really, it's a lot of work, and it's less satisfying than normal. Plus I usually teach a field class. And so it was going to be really challenging to do a field class, where I usually pack a van solid with people, and
Well, it is challenging for sure.
Dee, it's good that you're joining us. I owe you an email reply, and I'm very grateful for your patience. I'm actually working on it right now, so I promise you tomorrow you'll have a nice reply with all of your questions answered. Sorry for that.
That was for Dee? Yeah, all right.
No worries, Joachim, we're in Hawaii.
Yes, exactly. And Yoko has the best excuse of all to be late, because I asked his permission if I could tell this. He just got engaged last weekend.
Yay.
On the border of Lake Mead. Woohoo!
Yeah, it was a great time.
Good stuff. Yeah.
Congratulations.
Thank you very much. That means a lot. Yeah, I was mostly glad that I didn't lose the ring traveling to the mainland, so it was double relief.
Yeah. Yeah, it worried me that you kept it in your wallet too. I was like, oh man, you're just pulling money out at some point. It's gonna come out or
coins anymore.
Yeah, good. If you sit on it. All right, we'll give it a few more minutes here.
Yeah, often people trickle in a little late to the Pauhanas, so we'll see. Dan's here.
Dan, welcome.
Hello.
How are you?
Good.
Good. Your name came up today in a couple of conversations.
Good ones, I hope.
That's good. Yeah, I thought I'd have a dramatic pause there for you. All good.
Hey, Craig, long time no see. How you doing?
Nice to see you, Dan. Long time indeed. At least you got your brew.
Oh, I wish. I'm going on the raspberry lime sparkling water.
Am I the only one here with my proper quarantini? Does anyone else got one?
I just dry for a month starting today. So
yeah.
Yeah, you never know what's in this cup, right? It's just
it could be anything.
The cup could be anything.
Red wine out of a straw.
Great thing about water bottles, you bring them to restaurants and people don't question you, and you save lots of money on drink specials.
I will say, like, the American thing with not drinking beer on the street, but if you just have a brown paper bag around it, that's always been to me. Like, what could possibly be in there? Nobody knows, right?
Usually
that's an
lemonade.
extended metaphor for American thinking. Yeah.
Yeah.
We'll just put it in a brown paper bag, then they won't know.
Yeah.
The question is, though, like, the German way of doing it is just drinking the beer anyway, and everybody knows. So I don't know what's better. Just going to work in the morning at 8 a.m. and sitting next to a guy who's on his 5th beer. I don't know. Maybe the paper bag's better.
All right. Well, do you think we should get started with intros and stuff? And if some folks come on in later, that's totally fine. So thank you guys for coming to the Pauhana today. Today, our guest speaker is Dr. Craig Nelson, who's an associate researcher at Seymour, the Center for Microbial Oceanography Research and Education at Manoa. He's going to be talking to us about some really cool microbial stuff, using microbes to detect wastewater. So really excited to hear your presentation, Craig, and thanks for joining us. So do you want to go around and just have everybody say, just introduce themselves? And Yoko, did you have a question for the team?
Yeah, so we can do name and where you work, if you like, and then if you want to also add what the weather currently is like in the place that you were born.
Yeah. All right, you want to kick it off?
Yeah, absolutely. So my name is Yoko. I work for WAI as the project coordinator. My background is in wastewater engineering, and I was born in Neuss, Germany. And as far as I know, they're currently switching back and forth between slurry, snow, and very slippery ground. And I'll kick it over to JD.
Hi, my name is JD. I'm a student. I'm an undergraduate student at Waseda University in Tokyo. Besides that, I do a lot of freelance things that also don't have to do with waste. What was the other thing? Oh, I'm currently in Nuuanu, the Nuuanu area in Oahu. This is also where I was born. It's sort of overcast. I think it's supposed to rain tomorrow.
All right, thank you. All right, Dan, you want to go next?
Sure. Hello, my name is Daniel Amato. My day job, I am an environmental scientist with Element Environmental out of Pearl Harbor, Waimalu area. My night job is a researcher with the University of Hawaii / Surfrider, and I also am the Oahu chapter coordinator for the Surfrider Foundation's Blue Water Task Force.
Thanks. Roya, you want to go next?
Sure, I'm Roya Dennis. I work at Elemental Accelerator, and this is just hiding all the stuff on the bed behind me. And let's see, I was just looking it up. So I was born in Phoenix, and 57 degrees there.
Oh, and I wanted to add that when I was born in San Diego, it is nearly identical weather to Oahu. It's largely sunny and about 70, 72 during the day, which is, yeah, I guess that's cool.
Ideal.
Yeah.
Colder waters, that's all. Yes. Dee, how about you?
Hi, I'm Dee Fulton. I'm in Kona on the Big Island. I was born in Savannah, Georgia, and I haven't been back there since I was probably 15 months old. But it's 52 degrees there with a chance of flood warning. And over here in the Big Island, I serve on the Environmental Management Commission for just 7. Water pollution is an issue here because Kailua Kona is down there on Kailua Bay, and we've got really nasty aging iron sewer mains. Had a major sewer spill this summer, past summer, July 2020, 20,000 gallons spilled into the bay. But we know that there's chronic leakage because we know there's higher conductivity readings at the pump stations down there near the pier where people swim at the bay. So I'm really interested. This was a subject for conversation recently among my peers. And Dr. Rick Bennett's over here. He's a microbiologist, and he's made it clear to everybody, explained to everybody that you really can't correlate elevated enterococcus with sewer pollution. And that's well and good, and that's reasonable, but still, I think we have some health risks going on down there at the bay. And I'd love to know what we can do to get a better handle on that and get more information about what is the status, how much crap is leaking in the water or is not leaking in the water. I'd really love to know.
And I'll follow up that just because I'm from the sister city, Savannah's sister city, Charleston, South Carolina originally, and it's exactly the same temperature, 52 degrees. But I've been out here for a long time. My name is Stuart, and I'm the ED and co-founder of WAI. And Dee, that sewage spill, you said that's the one in Hilo in 20.
No, in Kona last summer. Yeah.
Last year, huh?
At a blowout. Yeah, there was a cement collar around an iron pipe.
Huh.
And the collar, the patch failed. Right downtown, right on the main drag, right on Ali'i Drive.
Wow.
Spilled right into the bay.
Wow. That's amazing.
Very visible.
Yeah.
Yeah. Thank you for telling me about that. I did not, somehow that escaped my radar. I don't know how, but yeah. Craig, how about you?
Yeah, so I'm Craig. Lived here about 10 years, and I'm originally from Southern California. I was born in Ventura County, and it is about 65 degrees there during the day right now. Sunny and warm, probably a lot like San Diego, but it's a little bit cooler there. We usually get a good marine layer. When I was a kid, I used to hate it, but sometimes now I kind of miss it, especially in the summer.
Yeah.
But yeah, you know, a lot of what I do, I was originally a freshwater scientist. I got my PhD working on, originally I was going to work on insects and, you know, as indicators of stream health. It's a pretty big deal on the mainland. And I got really interested in microbes once I started studying the nitrogen cycle. And I was like, oh, the microbes control all the nitrogen cycle stuff. So I stopped working on insects, started working on bacteria. And then I got my PhD studying lakes in the Sierra Nevada. So I became a limnologist and worked on bacteria in the lakes of the Sierra. And then for my postdoc, I was trying to stay, and I went to UC Santa Barbara for my PhD. And for my postdoc, I was trying to stay in the area because my wife was a student there. And so I became an oceanographer because you got to do something different. Otherwise, you'll never get a job. So I became an oceanographer and I started working on coral reefs, particularly looking at microbes in coral reefs. And then that's how I ended up here. So I'm in this building that's really nice called the Center for Microbial Oceanography, which Christina knows as well. And it's a really great place to work. It's like a big shared lab and everybody works on microbes. But most of the people here work in the open ocean. So they work out like at Station Aloha, north of here, about 50 kilometers north of here. And I was originally hired actually to work on seawater air conditioning and OTEC. And that didn't really become as big a thing as they anticipated when my job was advertised. So one of the first things I decided to do was study, I had lived on Big Island for a year in 2000. So I knew a lot about catchments and cesspools, and I decided I wanted to do coastally relevant stuff. So I reached out and started 2 local projects. And one was to look at cesspools and cesspool contamination of coral reefs in particular. And the other was to look at, I work with a guy named Neil Sims, who some of you might know out at Kampachi Farms. When I had lived on Big Island, I'd seen he has these like big nets out in the reef where they grow the Kampachi. So I just cold emailed him and I said, hey, you know, do you have any problems? Do you have diseases on your fish or anything like that? And at the time he was like, no, but I really want to start figuring out how we can grow limu. So we've been working on that for a while. So those are my kind of 2 big applied local projects. So yeah, so I think the reason Christina invited me here today to talk was just to talk about our work, trying to figure out what Dee is worried about, which is how do you tell when you have sewage contamination? And there's so many different ways, and we happen to do a lot of microbial gene stuff. So that's kind of our approach. So that's what I'll talk about today.
Great. All right. Thank you for joining us. And Christina will round it out with you and then make some announcements, and then we can launch into Craig's presentation.
Sounds good. I'm Christina. I'm the program manager at WAI. And yeah, I guess interesting, similar to Craig, I went from the SWAC to the sewage contamination in coastal oceans route, but it's just taken me kind of a long time. But yeah, I'm from Coopersburg, Pennsylvania, which is north of Philadelphia. My mom has been sending me pictures of like giant icicles all week. So I think that's what's happening over there. I think they haven't left the house in like 3 weeks or something. So, yeah. And then Yoko, you had a couple things, or Stuart, did you want to make an announcement?
Nope, I was going to pivot to Yoko. So
Okay.
Yeah.
Thank you very much. Yeah, so we do this every month. This is called a Mentee Poll, which is a live poll, interactive but anonymous. So if you would please take out either your phone and go to mentee.com using the code and the numbers up on the screen right now, or you can alternatively just open another tab on your browser. And then it's sort of 2 questions. It's very quick, but just getting some interactive feedback. And the first question is a ranking question. Rank these components in untreated residential wastewater by their typical concentration, first being the highest. And you can sort of drag it in the order which you believe is true. And then once everybody has voted, we'll move on to the next question.
All right, and you just have to put in the code at the top.
I use this when I teach a lot. It's a nice little program. But man, you got me stumped on this question.
All right, so in total we have 6 people on the call. No, we have more. We have 8 people on the call. So we have 6 responses, 7 responses now. Either there's one person will give the last person a little more if they want to. If not, that's fine. Give it maybe 5 more seconds. There you go. So currently your answers are nitrogen most, then BOD, then TSS, and then phosphorus. It's not bad. So BOD usually comes in as first, which is around 300 to 400 milligrams per liter, or also called parts per million. Then we have total suspended solids, usually at second place, coming in at around 200 to 300. Nitrogen is talked about a lot, which is why I assume people have ranked it here first. Nitrogen comes usually in around 50 milligrams per liter, 50 to 100, and then phosphorus usually 10 to 50 is fourth. So good job, everybody, very close. And then the second question is should just pop up on your phones on the same tab or whatever you had open. And the question is, what organisms involved in water purification can you think of? So literally any organism, anything that's alive that is purifying water. Doesn't have to be wastewater, but it can be if you want it to be. Wow, y'all are good. Somebody will have to teach me what desulfovibrio is.
I have a feeling that's Craig. Someone who knows that what they're talking about. In depth.
Right. Wow, still results coming in. That's great. So it looks like 2 people said algae, and then we have whales, we have bamboo reeds, aerobic bacteria, anaerobic bacteria, oyster and oysters. So I count that as twice as well. Grass, clams, sponge bacteria, and who doesn't know them, desulfovibrio. Thank you all very much for taking part in this Mentee.
Thank you. We just have a couple of quick announcements and then we'll dive into the presentation. Wanted to mention that we're tracking a bill, Senate Bill 369. It just passed out of the Agriculture and Agriculture and Environment Committee in the Senate today. And basically what that does is super important bill for the state to be able to make sure that homeowners are converting their cesspools. Right now, the only tool that they have is if you want any kind of permit to remodel or do an upgrade or anything to your home, then they can say you have to upgrade your cesspool before you can get the permit. The bill that we're focusing on now would allow that anytime you sell your house or transfer it with certain exemptions, that, when there's money on the table, that's the best time to upgrade the cesspool, especially with 30-year mortgages, because they've got to, by law under Act 125, convert all cesspools by 2050. That's Act 125. And so with a 30-year mortgage, we run up against that now. So this looks like it has a really good chance of passing, but if you want to support, please let us know. You can go on our Facebook page, or you can just email us. We'd love to have your support on that. And it's only got one more committee on the Senate side, Ways and Means, and then it'll flip over to the House. And it got pretty far last year. And then they inserted some language into it today that just allows people to upgrade their cesspools. Under the current law, it says you can upgrade to septic systems or aerobic treatment units. But we're trying to introduce new technology and make more room for innovation and affordability. And so they're just changing it to say any new system that's approved by DOH. So it just gives more room for that. So that's a quick update. If there are any questions.
What's the number, and can we do public testimony yet, or do we have to wait until it's up in the House?
When it goes to, I'll put it in the chat. It's SB 369, and the Ways and Means Committee is the next hearing opportunity.
Okay.
So that's WAM, and that'll be coming up, probably in a couple of weeks. Within a couple of weeks. Yeah. Thanks for that. And then other bit of news was we had a really successful town hall meeting. We're kind of going around the state, especially starting with priority areas across the state, kind of educating and getting input from communities about cesspools. So we did Kahaluu last month, and we had about 98 people. It was huge, and really a lot of engagement from folks. And so our next one is going to be on Wednesday, March 3rd in Waimanalo. And that's where Dan has done a lot of research and a lot of work, and getting kind of tracers with algae, using LIMU with the delta 15 nitrogen. And so he presented to us about that a couple of meetings ago. And yeah, we're excited about that coming up. We're getting good attendance. And then if there aren't any more, does anybody else have anything else to share? Things that are going on? Roya works at the Elemental Accelerator, and they're always looking for innovative new companies and to apply to their new cohort. And so that deadline opens up next week and will go through to the end of March.
Yeah, thanks for that. And I also want to share that we have an internship program with companies in our accelerator. And those are 20 different roles across the country. And students can apply to those. We are looking for business, marketing, business strategy, communications, data analytics, operations, biological research in Washington for drone seed. I mean, there's just so many different roles. I'll throw the link in the chat for y'all.
Awesome.
If you want to share it with your students or other students that you know of. There's really only 2 Hawaii-based roles, but these are remote internships, so they are really open to Hawaii applicants. And that is one area that I have not seen a lot of traction, is getting Hawaii students to apply. So I'd really, really I encourage you to let people know. February 19th is when applications close.
Right. Thanks for that. And then I will kick it to Christina to introduce our speaker.
Yeah, I mean, I guess we've kind of done introductions and yeah, so I guess Craig, the floor is yours to go ahead and do your presentation. I'm excited to hear about your microbes.
Sure. Yeah, so you guys are a eclectic group. So I'm going to probably try to skim over some stuff that you probably know like the back of your hand based on what you've all talked about and who's here. So if you want to talk about anything that I'm skipping over, just make me stop. And if there's a little time, I can pull up other things that I didn't prepare for slides or whatever. You know, I didn't really know what to prepare. So I'll probably talk a little bit more and have a little less visuals. Can I share my screen? Is that straightforward? Okay.
Yeah, should be good to go.
Let me do this. And let me do that. Okay, you guys cool? You can see it nicely?
Yes.
All right. So like I said, a lot of the stuff that we do as like an academic lab is really focused on natural coral reef function. So trying to understand all the, you know, hundreds to thousands of microbes that are in coral reefs, and particularly how they're different from the natural ocean around them. We spend a lot of time trying to convince people that coral reefs have their own microbiology. And that's important for the question at hand, because when you're worried about contamination of reefs from groundwater or contaminated groundwater or sewage runoff or whatever, you kind of need to know what's normally there. So I think this has been a reason that I've kind of maintained the focus on cesspool contamination and submarine groundwater discharge, is because it's good for people like me to be paying attention to this, because I think so hard about what's naturally in reefs. And so that way, if I see something out of the ordinary, you know, just from my own intuition or experience, I can say that's unusual, and it means there's something there that shouldn't be there. Whether that's from sewage or not is always the hard part. And that's really what Dee asked about. And so Dee, I'll try to make sure I answer your questions. You guys know this. There's a lot of cesspools on Oahu. There's a lot of cesspools on the Big Island. And that was originally how I got interested, was just the realization that a lot of my friends on Big Island didn't know where their cesspools were. On Oahu, it's even worse, I found out. A lot of people don't even know they have cesspools. So, but this is old hat to you guys, but there's a lot of cesspools. And they're concentrated in certain areas. There's a pretty important political divide. There's a lot of weight that goes along with having a cesspool. Whether you have a cesspool or not can be the divide between town and country. And so it's not so much that everybody wants to convert to sewer, right? So that's why what you guys are doing is really great, because it's great to give people an opportunity to have an on-site sewage disposal system that is not a cesspool. But a lot of what I work on is submarine groundwater discharge. So do you guys all know what submarine groundwater discharge is? Okay, I see a lot of nods, so I'm not going to go too much into it. This graphic is from Mauna Bay, right near here. That thing in the middle is the Kahala Hotel, which actually has a little mini seawater air conditioning unit in it, and that's why you see that plume of cold water coming out. And we're actually a little bit worried about that system, but that's kind of an aside. But submarine groundwater discharge comes out in a lot of spots in many places. Usually they're coastal. So even on the Big Island, a lot of these areas, the discharge of submarine groundwater is usually really close to shore. Sometimes it comes up in the reef. Sometimes it comes up along the sand, but it's usually not really far out until you get to these deep aquifers. And you guys may have heard Eric Gaidis's work on the deep aquifers on Big Island. That's a super cool thing. Totally different amount and volume and source of water, but a really cool thing. But submarine groundwater discharge has the potential to influence reefs quite a bit naturally. They're used to getting submarine groundwater. They're used to getting fresh water. That fresh water almost always had nutrients in it. So we think reefs are adapted to receiving some amount of fresh water, especially in Pacific islands like Hawaii. And so that's something that we study on a lot of islands throughout the Pacific. But here, the groundwater has the potential to be contaminated. So that's what I'll try to talk to you guys about today. So any questions about SGD? All right. And you're all super familiar with this. There's a cesspool. I usually try to inform people that a cesspool is a pit in the ground where your waste goes. And there's not a whole lot to say about it. And a lot of people are really surprised that cesspools are so simple, that they don't have a bottom, that they barely have a side, that sometimes after a while they don't have anything anymore. They're literally just a pit. And sometimes they're full, and sometimes they flood out. But yeah, we have close to 100,000 in the state, and a lot of them need updating. You're supposed to do it every 5 years. You guys probably know a lot more about cesspools than I do. I've actually never I've collected samples out of them, but I've never really looked in a cesspool. And I want to plant something in your brain for afterward. When the pandemic started, we got really interested in the idea of surveying sewage, because we work with a lot of groups around the country that were starting to survey sewage systems for COVID. And one of the things that we brought up was the potential to survey cesspools for COVID. And we thought about this for a while, and we're like, well, there are pump trucks that go around and service cesspools, but we basically decided that we didn't think they did it enough to get a good signal. So if anybody has any input on that, I'm keen to hear about it, because it's something, it was kind of an equity issue, right? A lot of people aren't on sewer, and detecting COVID when you're not on sewer can be challenging. We were kind of hoping to do it through cesspools. So you guys might have some input on that. So the big question that we try to deal with is when communities come to us and they say, look, we're worried. We think that there's something going in the reef. We don't know how to tell. Everybody has an opinion. A lot of people are like, oh, your cesspool is leaking. This person's cesspool is leaking. Oh, there's that group up the hill. They're the problem. So we primarily work, we've worked with a variety of communities over the years. Kahalu'u that you guys brought up. I'll show you some data from Puako on the Big Island D. We work right now in Hilo, Keaukaha. We've worked a lot in around Black Point in Monolua Bay, because a lot of those homes, despite their high prices, are on multiple cesspool units per home. And there's a variety of areas where we think we probably need to do more work. North Shore of Oahu is probably a good one, and the larger North Kaneohe Bay area. And so, yeah, we're always bouncing around trying to get little pockets of money to work on different areas. So if you guys have any questions about any particular area, I have a lot of data on various places that I'm happy to talk to you about and banter about. So any questions so far? Okay, so you all understand this. You understand subterranean estuary and submarine groundwater discharge and how it gets into reefs. An important thing to note is that when you do have some kind of sewage contamination, you almost always have nutrients. And Yoko did a really nice job with the Mentee, illustrating that one of the main things that's in sewage contamination is nutrients, nitrogen, phosphorus, BOD, which is a weird engineering way to talk about organic carbon, right? So BOD is actually organic carbon being used and respired to use up oxygen in aerobic systems. but VOD is organic carbon. And then the other important thing to note is that when we have nutrient contamination of reefs, it could be from a lot of different sources. So one thing that we harp on a lot is that just because you have nutrient contamination in your reefs, you have algae, something like that, doesn't necessarily mean that the source is sewage. Groundwater already has a lot of nutrients in it. And there's also lots of other sources of fertilizer, as Dan knows super well and has worked on for a long time. Okay, so I'm gonna really quickly get to what we do. So how do you tell if whatever is going on to make your reef different is sewage? And an important thing that you guys, I think, all know, but it's worth saying out loud, is the things that the microbes that we look for, Enterococcus. In fact, somebody mentioned it at the beginning of this call. Like Enterococcus is not, Rick D, you brought up Rick saying that Enterococcus is not a good indicator of sewage. And that's true. I mean, sometimes it is, sometimes it isn't. It was developed not here. So, but all of these things are indicators. They're not inherently dangerous themselves. So this little plate here is a plate designed to detect, growth of cells of, I don't know if this is an Enterococcus or a Clostridium plate. And we also do this with a variety of E. coli detectors and stuff like this. But none of these organisms actually hurt you. They're just somebody somewhere else figured out that they correlate really well with health effects. When people get stomach aches from swimming in a lake, there's a good correlation between the things that'll grow on the plate and those effects, but it doesn't mean that they're causal. It just means because usually the things that actually make you sick are super rare and hard to find. So what you're looking for is indicators. So this word indicator is really important. And so the traditional ones that we use are Enterococcus, and this plate here is what's called an IDEX tray. You guys have all used these. Christina, you're probably intimately familiar with these. They're a really quick and dirty way to basically mimic what's on this plate. Each one of those wells, in theory, has one of these little cells in it, and they're growing and using up the indicator. So this is kind of a cartoon that my graduate student made about culture-based indicators. So she had a really good time making this, so I figured I'd use it for you. So she goes down to the water with her little tube. She gets her little plate. She puts the water on the plate, and miraculously, these things grow on the plate. The things that are growing on the plate here, at least regulatory wise, are usually Enterococcus, and we usually do plates for a thing called Clostridium perfringens. And I can talk to you guys about why we do that and what it means and blah, 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 if you're interested. If they grow up, then you have a measurement that has a regulatory framework around it, and you can say, yep, there's something going on here. Now, the big problem with this is that these indicators weren't developed here, and they're not very good. They were developed a long time ago. They're based on old ideas of microbiology from the 1980s and 1970s, and they're just not the best. So we need to come up with better tools. And our big thing is to look for other bacteria. In particular, my interest is in the idea that Hawaii is fundamentally different from the mainland United States. It's fundamentally different from most first world countries. So there aren't a lot of tropical rich countries. And Hawaii is an example of one. And we have a little bit of a responsibility to the rest of the world to think about that the ecology of our diet and the ecology of our ecosystems is different here. And all of these indicators are developed in Cincinnati, Ohio. Cincinnati, Ohio is where the EPA is a big lab and they work on indicators. And what do people eat in Cincinnati, Ohio? It's a Mentee question.
Meat.
Meat. And what else?
Beef hot dogs.
Hot dogs.
Potatoes?
Potatoes. Totally. So Dan, what's the isotopic composition of a person from Cincinnati, Ohio, or their waste?
Definitely about 6.325. I'm pretty sure that's it.
So the curious thing about people from Ohio and all the indicators in the Great Lakes, which is where all this stuff is done, is they're like 90% corn. Everything they eat is corn. All the meat they eat is from corn, fed on corn. They eat a lot of processed food. It's all made from corn syrup. Their isotopic composition and their gut flora and their sewage is all derived from corn. All the microbes in their gut love corn products. You go here, we don't eat that much corn. We still eat a fair amount of corn because we eat a lot of processed food, but we eat a lot of rice. We eat a lot of fish. We do eat a fair amount of like beef, to the extent that we eat beef that's grown locally. It's probably fed on grass with some corn. But the bottom line is our diet, if I were just to guess, is probably at least half as much corn as where these indicators are developed. And these things that I've pointed to, so this is a tree. If you guys have ever seen a phylogenetic tree or a tree of life kind of thing, this is just a tree in a circle. And all the little leaves on the tree are genomes that we have of bacteria. This is old. This is like 2007. Now we probably have, I don't know, 6,000 or 7,000 leaves on this tree. But each one of them is a genome of a microbe that we figured out. We're always finding, we know there's like 30 phyla of bacteria now. We're finding new ones all the time. And the three that we use as indicators are flagged here. So the one at the top is the Enterococcus. Sorry, no. The one at the top is, sorry, I've got my thing in the way here. I'll move it. The one at the top is E. coli. And this whole family is full of microbes that we find in our guts. The one in the middle is Enterococcus. So that's in a group called the bacilli. And the one at the bottom is these things called Bacteroides. And if you've ever read anything about like gut health, Bacteroides is one that is really important in our guts and the guts of most animals. So our goal, and if you get nothing else out of what I'm talking about, is in line with the goals of a lot of people around the world, which is to find types of microbes that genetically tell you where the microbes came from. So this group down here, the Bacteroidales, it's a weird name, but you can get a Bacteroides out of your gut, and out of the gut of a cow, and out of the gut of a chicken, and out of the gut of a pig, and out of the gut of a bird, and out of the gut of pretty much any animal. And you can tell them apart based on their gene sequence. So if you get a sample of water out of the coastal ocean, and you look at the Bacteroides genes in it, you can tell if the Bacteroides came from a cow or from a person. And this is pretty well worked out. We've been doing this for like 15 years. And I've done a lot of it in the Eastern Sierra where we, have these like big, like litigious fights. You'd go out to these streams and there'd be cow patties sitting in the streams. And you'd go to the farmers and you'd be like, you guys need to stop letting your cows walk in the streams because it's raising our fecal coliform counts. And they'd be like, not us, it's the deer. It's the deer upstream. It's not our cows, definitely not our cows. And you could be like, look, there's a cow patty right here in the stream. And look at your cows standing in the stream. They'd be like, no, it's the deer. And so we had to figure out a way to differentiate deer gut microbes from cow gut microbes, from people microbes. And so this is all well established. And so we call this microbial source tracking. And it's fancy. It uses gene sequencers, costs a lot of money, requires some expertise to make sense of, but it's cool. The EPA is on board. They've come up with their assays, and I'll show you a couple of those at Puako in a second. But just so you have an idea of how it works, You basically get your sample of water, you take the DNA out of it, you amplify it by this thing called polymerase chain reaction, you sequence it, and then you apply some fancy computer stuff to figure out what microbe is there. And to go back to our cartoon, the way this looks is instead of, pouring your water onto a plate, we literally just push the water through a filter, and we take the filter, and we remove the DNA from it, and we sequence that. And in theory, the difference is that instead of the plates are selective. So they have like an agar on them, and only certain things grow on that agar. But the DNA is not. You can basically sequence all the DNA and you can tell every organism in there. And my big point is that if we have unique organisms in our guts and our sewage that you wouldn't find in Cincinnati, and the EPA would never develop markers for, because we eat fish and we eat rice and we have different lifestyles and we have coral reefs instead of Great Lakes or the Delaware estuary or whatever, then we can find organisms that are indicators of sewage in Hawaii that are specific to Hawaii, and maybe tell us something about, what kinds of, based on what kind of microbes we know are in reefs naturally, and what's in soil, we can then look and find microbes that are, my student here has kind of circled these ones, but the idea is those little orange guys on that cartoon would be something that is an indicator of the guts of humans here based on our diets. So yeah, that's all I'll talk about conceptually. And so I'll stop for a second and ask if you guys have any questions before I talk about Puako a little bit. Yeah, go ahead, Dee.
Yeah, I have a question. I think we have our equivalent of deer here in Kona, and they're called the homeless people. So if we get poop and we know it's from a human, yeah, but the homeless, they're pooping down there. And so, and when you do these assays, it reminds me of phylochip.
Yeah.
And you can't really you're not getting a quantification of the level of bacteria in the water, are you? So it's a great in theory, but it has its limitations. And that's what I'm thinking.
Yeah, so the phylochip is an old thing that was developed by Berkeley Labs, and I worked on it about, 15 years ago. And that's just a percentage. But with these, we actually do get counts. So these are direct counts of the number of cells that are from, like I can count the number of gene copies I find in a sample that came from a person.
Ooh, that's exciting. Okay.
If it worked, it would be exciting. Unfortunately, the assays that we're using right now are from Cincinnati, so they don't always work, but sometimes they work.
Craig, can I just jump in here? So you can quantify them, but correct me if I'm wrong, but I don't believe that's possible if you're doing more of like a higher level sequencing, where looking for many organisms at once. If you're trying to identify many organisms, like the full community, if you will, you cannot do quantification. But if you've singled out a few that you actually do want to find, you can quantify those.
That's exactly right. So the idea, a lot of our like more basic research is to use these whole, like the phylochip kind of thing that you brought up, Dee, where you look at everything agnostic to what you're looking for, get this idea of things, in places where there's contamination or places where there's no contamination, get an idea of things that are different between those two places in percentage, and then design primers. We call them primers, but basically they're just little things that allow us to do the polymerase chain reaction, but those will target a specific organism, and then we can get quantitative data on that. But you're exactly right, Dan. You have to know what you're looking for to get an actual abundance of it, at least right now.
And that being said, are you able to do multiplexing or look for more than one bacteria or organism in one sample tube?
Yeah, that's super easy. The limitation is which ones to look for. not the chemistry or the genetics. That's, you can look at hundreds at once, no problem.
Gotcha.
So we have kits available, but they haven't been adapted to Hawaii yet.
No, the real shortage is there aren't a lot of specific markers. It takes a long time to develop a marker. So the EPA has certified one that is a human specific marker. If you find it, it means that there's probably human sewage there. It's not from any other closely related mammal. And what would be ideal is if we had 20. And what would be even better is if we had 20 that were like mainland developed and another 10 that would kind of capture, variation that we find here locally in diet or something. Because you have like 300 or 400 dominant bacteria in your gut right now. So any of those would work fine. And the difference between your gut and Dan's gut is an important thing to capture. And particularly when you have a diverse population like we do here, big diversity of socioeconomic level, big diversity of diet based on all kinds of like cultural mixing and stuff like this, we need a pretty broad swath, and none of that's been developed. And we're not really in the business of developing all that stuff, but that's what we'd like to see happen. And that's kind of what we're doing as our first pass in a lot of these systems. Kahalu'u, the Alawai Canal, Keokaha, and Puako are kind of our targets right now.
JD, it looks like you raised your hand.
Hi, yeah. I was just wondering if there have ever been issues using these markers developed in Cincinnati in other parts of the mainland, or are their diets just sort of similar enough that they haven't had an issue there?
Well, it's a really good question. So I'm just speculating as to why they don't work super well here, because the observation empirically is that even in spots where we know from multiple other lines of evidence, including culture plates, Delta N15 work like Dan is super good at, and observations like in some places, like in Puako, we've put dye in people's toilets and flushed them and watched it come out 6 or 8 hours later. So we know that there is some kind of, and the markers aren't really lighting up. And I speculate, based on a lot of past work on gut microbes of animals and things, that the reason is because these are so specific to a particular place and time. And Cincinnati is very different from here, but I don't have evidence for that. And I will say that most of my colleagues who work in areas around the mainland, mostly on the East Coast and around the Great Lakes, they have no trouble. Like if they have an area where they know there's a crack in a sewage pipe or there's intrusion into a sewage pipe, these things light up like crazy. And I think that has a lot to do with the fact that, one, they're looking at sewers, not cesspools, which have a different ecology. So cesspools are fundamentally different ecosystems than sewage drains are. And the other thing is diet. And we know diet has a huge effect on the types of microbes you find. If you look at people around the world, they all have vastly different microbes in their guts. And the mainland is relatively homogeneous, compared with Hawaii. So I just am speculating. But that's a really good question, JD.
I see. Thank you.
And this is kind of my, what do you call it when you stand up on your, like, I want to say high horse, but not high horse, but like
soapbox.
Soapbox, yeah, this is my soapbox. Like, I don't have the resources to develop these things, but the federal government does. But I really want the federal government to recognize that when they develop something in one place, it doesn't mean it's going to work here. And there's a history of that here. There's a lot of microbiologists who came in the past who also pointed this out. One of the reasons they pointed it out was like the old culture-based metrics often were really high in Hawaii. And they'd look for like Enterococcus and stuff, and they'd be like off the charts. And the EPA, then the federal level, was like, Hawaii is disgusting. And so people were like, no, Hawaii is not disgusting. It's the tropics. It's not the Northeast. So there's a reason we have a lot. These microbes are not specific. These plates, what's growing on the plates, they're not specific organisms. And so we get really high counts just because we naturally have a lot of these organisms in our soils and in our water because it's always warm here. And so it was kind of the flip problem. It was like we had really high numbers and we needed to explain that that's just naturally how it is here. It's just different. And so we are now facing this issue where the things that the EPA is developing as these specific markers are just not picking up what we know is sewage pollution. And we think it's because they're looking for the wrong things.
Okay, just a heads up that we're at 6:30 right now, but we started a little late. So if you want to do maybe a 5 minutes wrap up of any other important points you want to share, and then we'll make sure everybody can get to their dinners and evenings.
Yeah, I mean, I just wanted you guys to kind of see what this looks like in a place where we kind of know what's going on. So we've worked in Puako for a long time with our collaborators at Hilo and with Manoa and a variety of other folks. A lot of you may know Courtney Couch and Becca Most and Tracy Wagner at Hilo. And one of the things we did is this is an area where we know there's some sort of very near shore sewage contamination, debatably low levels. But if you're not familiar with this area, it's up north of Kona, and it's a pretty small area with a bunch of houses that have cesspools. And are looking, this community is very interested in changing that. And a lot of you probably work with them. But what I wanted to kind of show you is what this data looks like. So this is Delta N15 data, which you're all familiar with because Dan probably talks about it a lot. And these are pretty high numbers. They suggest that there's some sewage, some human or animal associated sewage contamination here. And there's a fairly high numbers of the state regulatory approved, these IDEX tray kind of things, Enterococcus, which don't work great as indicators here, but are okay. And so that's another line of evidence that there's some sewage or some kind of microbial contamination here. Clostridium perfringens is a supplemental microbe that we use here in the state to sort of certify that the Enterococcus is not just from environmental sources. And the Clostridium perfringens numbers here are pretty high in certain areas. And what we wanted to do was kind of be like, okay, you guys understand this place pretty well. Let's go in and try some of these markers that the EPA has developed. These should work really well. And this is the data from the EPA markers. So we had two different markers here, and they have stupid names, HF183 and back hum. It's like a terrible name for something, but they're genes from organisms that are thought to be indicative of human sewage. And this, out of any place that I've worked in the state, this is where we get some of the highest numbers here. But they're basically really low, 1 cell per 100 mils. So like if a person from the Great Lakes looked at these numbers, they'd be like, oh, you have no problems. You have nothing going on here. In a place where we have high Enterococcus, high Delta N15, high Clostridium perfringens, these are all state things that we understand for sewage contamination. These numbers should be like off the chart. They should be like 100 or 500 or 10,000 or something like that. And they're really low. And it probably does have to do at least somewhat with diet and with the source, which is cesspools and not sewers. So, but you can see this pattern. You can see that the technique is working, even if it's not working exactly the way it should. And so I just wanted you guys to get a feel for kind of our attempts to validate these methods that are developed elsewhere. And what we're trying to do now is go into these whole community surveys and be like, okay, there's 1,000 microbes here. what other things are we seeing that actually do show big differences between the contaminated and uncontaminated areas? And so everywhere we're looking for these kinds of things, a place that we know is contaminated or we suspect is contaminated, and a place adjacent that we don't think is very contaminated. So anytime you see that, let me know, because it's an area where we can go and try to develop these markers better and find new microbes that are associated with our guts that might be good indicators of sewage contamination. That's it.
All right. Thank you, Craig.
So yeah, any other questions about any of this stuff?
Well, oh, sorry. Good.
You can go.
I was just gonna say, I would like to follow up with you about some of the work you've been doing in the areas you mentioned early on in the talk. That a lot of them overlap with the DOH priority areas, and I'm curious to see what kind of data that you guys have collected there. Yeah, that's I'll follow up with you later because we're kind of at the end of the talk. And Dan,
Sure.
why don't you go?
Yeah, I just wanted to say, Craig, you'll probably be interested in some results I'm writing up right now from our statewide wastewater survey. We did work on Hawaii, Big Island, Maui, and Oahu, looking at using modeling and coastal collections of seaweed to determine where we believe priorities should be placed. This is for the legislature, so that should be of use. But I wanted to say that I think one of the, correct me if I'm wrong, maybe I don't have access to the studies, but I think one of the biggest data gaps that we really have in this whole, surrounding all the questions that we still have to answer is, what is transport from a cesspool through the aquifer and the ground look like in the different types of geology that we encounter? I mean, obviously at Puako, we're looking at very young lava rock. It's very porous. There might be lava tubes, and that's why you have a 6-hour travel time from a flush to a reef. Now that's unusual for a place like Oahu, or probably even Maui. And so I really would like more information on what types of viruses and bacteria are transported, how far, what their lifespan is underground. I think that would really, maybe Yoko has got access to some of this kind of more engineering science stuff. But I feel like that's a big missing piece is if you have a cesspool in the Waimanalo beach lots and you're going straight into sand, is sand a filter? I mean, it is for pools. Why is it not for a cesspool? And so I think there's a lot of questions we still have to answer that are kind of basic, almost engineering style research questions that could be done in a lab. And so that was kind of one point I wanted to bring up. The other was, Craig, what's your experience with inhibition in your qPCR assays? I know Eric Kears has been doing similar work. He's actually been working with the state to develop some sort of microbial risk assessment. And from what the DOH is telling us is that they have trouble finding pathogens and it's not working. And so there's these inhibition issues, and there's the fact that they just can't find pathogens that are reliable to create a new indicator. So just what's your take on inhibition with qPCR and the availability of some of these pathogens?
Yeah, I mean, those are hard, long questions to answer. The first one, I'll tell you, there is a lot of work done on sand. There's a woman who grew up here, Allie Beam at Stanford, who's spent the better part of the last 20 years studying sand beach filters. So for the Waimanalo work, I think it's really good if you either first look at her papers and then probably talk to her. She's from Kailua.
B-A-M?
B-O-E-H-M.
Okay.
She's been at Stanford for a long time, but she grew up here, and that sand question and more generally the idea of transport and how long the microbes live and whether their genes are there when they're dead and blah blah blah. These are all really important questions. A lot of the work we did in Kahalu'u was in the streams to try to avoid the long distance problems, was to try to look at where the cesspools are directly contaminated in the streams and then kind of track that down the watershed. So that first thing is yes, that's an important priority area, and it's a big problem for pretty much everywhere. So it's something that, I think Hawaii can get money to work more on that. And Merrick is a great person to work on it. This question about the pathogens is a different one, because Merrick has been looking for pathogens, the things that actually make you sick, as opposed to indicators, which are much more abundant and are predictive, but are not the things that actually make you sick. It's really hard to find pathogens. You got to filter hundreds of liters of water or go through, 1,000 tons of sand to find, the 10 viruses that can potentially make you sick. It's not that they're that rare. It's just that they're really hard to find. And you want something that's really abundant, that's a good predictor of the rare things. And, PCR inhibition, these are all technical questions. We don't really see PCR inhibition much. It's not really that big a deal. Probably it's the assays that aren't working terribly well because they just aren't designed for here. There's no reason they should work well here. So we have different guts.
Yeah, you're saying the primers aren't great.
The primers aren't great. You don't have the Bacteroides that they're looking for in your gut. These aren't the Bacteroides you're looking for, right? You have something else in your gut because you don't eat the same thing that the people who these were developed for eat, in my opinion.
And it's great to see Dan and Craig talking about these things, because this really is high level. We're sitting in on like two of the best researchers in this topic. So we're really stoked that you could join us, Craig, and share your research. If you go to our website, waicleanwater.org, WAI cleanwater.org, you can see the most recent Voice of the Sea episode, which featured Craig Nelson's work. There's a great video, and then one before that that Dan is featured in. So there's a lot of good other resources about some technology that we're introducing. Our new Cinderella toilet,
which my daughter totally wants. She's like, "Papa, can I have one of those?" And I was like, "Sure, we'll put it in the backyard."
Stuart, or Christina, whoever the moderator is, I got a question. Dan brought up a really good point, about the filtration rates. And over here, the County of Hawaii requires new construction to use a septic tank and septic field. And they're not saying anything about any any nitrogen absorbing sawdust or any other carbon source. And I have a concern about that. It's like, gee, people are having to spend all this money on digging out in Blue Rock to put in a septic field. And is it really going to be much different than a cesspit? So, and yet I spoke to a friend of mine up in Waimea, and he said, well, we got lots of dirt up here. So how does the county go about drawing the line about where to put in which alternative? Or is the Cinderella toilet going to be so cost efficient that we don't have to think about septic fields anymore?
Yeah, that's a great question, and requires kind of a lot more answers. This Cinderella incineration toilet is going to be really good for remote areas where they're or areas that are where if you just need a toilet and you don't have all the gray water in the showers, everything else, it's the perfect thing, off the grid. You don't need, so we're talking about $4,000 versus minimum $20,000 if you're going to do a leach field, septic tank, and the whole thing. So in that sense, there's no comparison. It's going to be much cheaper, and you're guaranteed 100% pathogen free. And so that's the real advantage with incineration toilets like Cinderella. But because in the United States and many developed countries, we don't separate our black water from our gray water, it's more, we have large volumes. And so you have to treat that. And so that can be really tough because you do need a larger septic tank, and you need a leach field to help treat it. So, but yeah, we'll talk more, and there's a new video, Dee. I don't know if you saw on our website about Cinderella, but you can look at some of we posted it on our Why Clean Water website and check out.
I think the last video I watched was Yoko talking with, I can't think of the entity, but it was really well done, Yoko.
Yep. Yep. Thank you. He did a good job on that. Appreciate that feedback. But we usually try to keep it to an hour, so we're 15 minutes over. So I think we'll wrap it up. But really want to thank all of you for attending, and especially Craig. We really appreciate your presentation and all the good research you're doing. And JD, we're glad you could join us, our newest member. And to the rest of you, we'll see you next week. It's always the third, we switched it, so now it's the third Wednesday from 5:30 to 6:30. And if you have any ideas, or if anybody wants to present, please let us know. We don't have a speaker for our next one, and we're always looking to have really cutting edge research or just interesting topics. So let us know, and feel free to invite friends.
Cool. Thanks, guys. It was great.
Thanks.
Yeah.
Bye.
Have a great night. Aloha.